Okay, so GIT motility. Within the digestive system, we find that multiple processes are involved to allow that food travels down the digestive tract in a controlled manner. It will involve both the types of muscles that we know, skeletal muscle, and this is usually the skeletal muscles are involved at the beginning of the GIT and at the end. So on both ends of the tube, you have skeletal muscle input. And then in between, we have smooth muscle input. They're the ones which determine what type of movement that we are going to experience within the tract. So the other thing you need to know is that if you recall when we were talking about regulation, we say that we do have the enteric nervous system, which usually has that local control just within the area where the food is. But this can be modulated by the autonomic nervous system, and of course, with the autonomic nervous system, we also talk about the peripheral nervous system. So we have the peripheral nerves, they are going to work through the autonomic nervous system to control the enteric nervous system. Now, for that movement, apart from the neural control, we can also have hormonal inputs. For instance, we say that we have things like gastrin, which can initiate movement in the stomach. You can have motility. Motility also initiates movement. Then we have things like somatostatin, which will inhibit. Among the processes that you have to discuss when you're thinking of movement, it will include one, the chewing and swallowing component, and this is involving the skeletal muscle. It involves parts of the mouth. It involves the initial part in the swallowing process. Number two, you have peristaltic movements, which are mostly propulsive movement. They move food in a distance towards the arboreal region from the oral region. We have churning or mixing, and this happens in several areas of the GIT, which include mostly the stomach and the small intestines. Then we have segmentation, and segmentations will discuss what it is, and you're going to see a lot of segmentation either in the small intestine and to an extent in the large intestine. Then we have another form of peristaltic wave, which is slightly different from the peristalsis that you know. And this is the one that allows, if there are sweeping movements, whereby you have contractions of movements which start from one end of a sphincter and they end up in another end of a sphincter. And these are the ones that we call the migrating motor complexes. Then we have mass movements. Mass movements are the ones which help in the movement of contents within the colon, and then they end up leading to the process of defecation, which is the final step in the movement process of the food within the GIT. So we look at all these processes and where they occur. So in summary, if you look at this diagram, it is summarizing the different regions of the GIT and the type of movement or functions that we find within that area. So within the esophagus, we have mainly peristalsis, which is propulsive movements. It acts like a conduit between the mouth and the stomach. Then the food enters the stomach, and the first thing that we say, it's a pocket to hold the food contents. For that reason, the stomach is acting mainly as a storage, but during this storage process, we have a lot of mixing and churning. So this food is always being thrown to and fro, and in that process, you are tearing it into smaller and smaller pieces and mixing it with the gastric juices. Now it reaches a point where some of the food is ejected out during gastric emptying, and for that to happen, we need the peristaltic or propulsive movements. Now, once it goes into the duodenum, between the pyloric sphincter and the colonoidal sphincter, you find that during this part where we have the small intestine, we have a lot of mixing, and the mixing process is allowed by a process called segmentation, which we are going to talk about. But in addition to that kind of mixing, we also have peristaltic movements which will propel the food in a distal direction. Finally, these food contents will get into the undigested material, that is, it will get into the colon. And the colon is another pocket that acts like the stomach because it will hold the contents, the undigested material, until they are ready to be defecated. For that reason, it is also acting like a storage organ. But within the colon, there is also that process of mixing and churning, where we see a little segmentation, and there is all this mixing so that it allows absorption, especially of water. And we also have the peristaltic or propulsive movement, which will propel the food to the distal direction. So the first stage, I'm not going to dwell into the details of swallowing because we did that when we are looking at the events that happen in the mouth. All I can remind you is that there are 3 stages. The first one being the voluntary stage, and the second one being the esophageal stage, the pharyngeal stage, and the third one being the esophageal stage. But one thing I have to tell you is that for this swallowing to occur, we need a lot of receptors. We need the voluntary input here to initiate the process. So when the food goes to the back, now we start the process whereby you bring in the component of the peripheral nervous system, because we have tactile receptors which will send information to the swallowing center and from there initiate the process. Number 2, I wanted to alert you when it comes to swallowing, always remember the upper esophageal sphincter and the relationship with the lower esophageal sphincter. So the 2, they hold contents between the esophagus. And 3, we have peristaltic movements which will push the food bolus to the stomach. It is also important to note that it is not all the food that enters or initiates peristaltic movement manages to go all the way to the stomach. So you may have something that you swallow, but with the initial wave of peristalsis, the food is stuck somewhere in the esophagus, and the wave of peristalsis continues and diminishes to the end. Now, what happens is because of the distension, yes, Asna?
Madam, Anastasia was kicked out, so she's asking if someone else could take over for her.
Oh dear. Anybody else who wants to be recording? Otherwise, we won't have a recording for this. Okay, I'll put all of you, Lillian and Ivy. I've given you the rights because I can see quite a number of people have been kicked out and coming back. So even though many have given you, so the more people who record, the better. Okay. Okay. So now I just wanted to alert you that when you initiate, you might have food stuck. And now when it is stuck in the middle, anywhere in between, within the esophagus, what happens is because of that distension around that area, you continue sending information to the brain center or the swallowing center. So as long as you have those afferent neurons firing and telling the brain that you have something that is stuck, it will send efferent inputs to initiate another wave of peristaltic movement. So when you have a second wave that is coming as a result of food that is already within the esophagus, we call that a secondary peristaltic movement. And I think that is the one thing I had not mentioned. So I won't go through these other stages because we had talked about them. But I just wanted to mention that we do have the initial peristaltic or primary peristaltic wave. And if food fails to get into the stomach, then you have to have a secondary. That's where we have primary and secondary peristalsis within the esophagus. Now, the other things I wanted to discuss is before I go to the next level or the next organ of the digestive system, which is the stomach, I wanted us to look at how do you manage to initiate this, whichever kind of movement we are talking about. For you to understand, I think we've done a lot on ANS, PNS, and ENS. So in the previous slides when we were discussing regulation, we looked at the role of all these nervous arms of the nervous system. Now, as for the smooth muscle, I will take you back to Physiology 1332, where we are looking at structure of smooth muscle versus skeletal and cardiac. So we need to remind ourselves what the smooth muscle looks like so that we can understand how it works. Number two, we also have bringing another component of cells known as the interstitial cells of, some people call them Kajal, others call them Kahal. But these are just satellite cells. I'll talk more about them. Then, of course, in the regulation, I also discussed about hormones. So if I leave this for the time being and I go to the smooth muscle, let us look at the structure just to remind ourselves, because I had shown you this structure when we were doing 1333. It tells you that smooth muscles are arranged in bundles, and some of these bundles are separated by some connective tissue. Have neurotransmitter vesicles. So anytime you depolarize this autonomic nerve, it's going to allow this neurotransmitter to be released into the surrounding smooth muscle tissue. And those that are in direct contact, they may have receptors for that neurotransmitter. And those receptors are enough to cause depolarization. But because of the gap junction, when that depolarization happens to the cells that are in the vicinity of the neurotransmitter, the depolarization will be transferred to even cells that are not within close proximity. So the whole of that area will act as a single unit. And if we are talking about a part of the GIT, remember you have the circular muscles and then you have the longitudinal muscles. So if you have depolarized an area here, you find that this depolarization may spread in a concentric manner, so all around the circular muscles. And at the same time, if you depolarize the longitudinal muscles, this depolarization will be transferred in a long region within that place. So all this area within that GIT will act like a single unit. So these two slides are just to remind you on the structure of the smooth muscles, showing you the innervation and secondly, the gap junction. From there, I will come to the second component that you need to understand. This is a new concept I had not introduced to you. So for those people who have a problem with attention, I would seek that you pay attention as I talk about the interstitial cells of Cont'D. Now, in the heart, we have what we call the pacemaker cells. And those pacemaker cells were very rich in little channels. And these little channels, or you used to call them the funny channels, they would cause a continuous depolarization of the cell membrane of the pacemaker cell. And this steady depolarization, if you are to draw a graph showing you the resting membrane, or what is supposed to be the resting membrane potential at that area, you find that the depolarization will move from the resting potential, and because of these channels, there is a continuous depolarization until a threshold is achieved. And when the threshold is achieved, we have that massive depolarization, and then it goes back to the resting position, and then it starts the cycle again. And these were what we called the pacemaker potentials. Now, in the smooth muscles, we have cells which behave in a similar manner. These are also referred to as the pacemaker cells of the GIT, but they do not behave exactly like the pacemaker cells of the heart. So for those people who are lost, I've mentioned a lot of pacemaker cells. Interstitial cells of Cont'D set the rhythm in the smooth muscles, but the rhythm they set is different from what we see in the pacemaker cells of the heart. For the simple reason, pacemaker cells of the heart always end up with an action potential when the threshold is achieved. Now, with the interstitial cells of Cont'D, we find that there is that continuous depolarization. I'll use a blue color. So if it depolarizes and it doesn't reach threshold, you find that repolarization occurs. Then it reaches the lowest point and starts again another series of depolarization. And if this does not reach threshold, again, resting membrane potential is being restored. And so what you'll end up having with the pacemaker cells in the smooth muscle is these continuous waves that you are going to see of depolarization. And these waves are what we term as the slow waves. Now that electrical rhythm of depolarization, repolarization is what we refer to as the basic electrical rhythm. And it is set in place by the interstitial cells of Cont'D. So let us discuss the Cont'D in greater detail. I don't know whether I have a slide. I'm not sure if I have a slide for that one. I don't. So there is I don't have that slide, but I wanted to show you. We have the smooth muscle cells. What you find that when we have smooth muscle cells like this, of course, we have the two layers. So we have the longitudinal, and then let's say we have on this side the circular smooth muscles. So if we are, these are circular smooth muscles and these are longitudinal ones. So what will happen is that in between these cells, we are going to have things that we call, they are star-like because they have many processes. And these cells are what we call the interstitial cells of Cont'D. We have some within the circular smooth muscle, and we also have those that are within They are all satellite because they are spreading out their processes, and you find you have quite a number within the longitudinal muscles. And these cells are connected to one another by gap junctions, and they are lying in between the smooth muscle layers. Now, if we take one of these cells, the interstitial cells of Cajal, their origin are not actually muscular tissue. Their origin, they are more of mesenchymal cells. They are the ones which form the mesenchyme during development. But then they become modified by having all these channels which will allow the slow leakage of iron from the interstitium. Now, please note the second difference between these interstitial cells of Cajal and the pacemaker cell is that here we have leaky calcium channels. So remember, in the heart we have the funny channels that allow sodium to go in. In this case, we have leaky calcium channels. Now, when these calcium channels they go in, they're going to cause depolarization of this membrane. And this membrane, remember, is in direct contact with some of these smooth muscles, but also with other satellite cells through the gap junction. So when we have this depolarization, it will sort of be, if you look at the electrical activity in that area, because of the continuous depolarization across this area, the whole of that place will have a continuous depolarization. And then if it does not hit threshold, you find that depolarization diminishes. And this is because the calcium that managed to leak in is usually also being pumped out of the cells. And therefore there is that repolarization. Once it hits rock bottom, it doesn't sit there. Again, the leaky channels take over and you have the depolarization. Now, if this depolarization crosses the threshold value, then the next thing we are going to see is there is massive influx of sodium and calcium, which causes what we call the spike potential. And this spike potential is the only one that can initiate a muscular contraction. So in this case, if you are to look at the tone of the muscle and you are looking at whether contraction of the muscle is occurring, you find that if you are to do an EMG, this one will be very straight because there is hardly any contraction. But when we have a spike potential, then we initiate contraction of the smooth muscle. So the smooth muscle contraction comes only after the spike potential. The spike potential comes when the depolarization initiated by the interstitial cells of Cajal reach the threshold value. So now, what is the threshold? Usually, by around minus 45 is when you can initiate a spike potential. Below that value, you find there is no spike potential. And the lowest depolarization we usually find is within the GIT is right around minus 65. So you find these BER that we are talking about are always fluctuating between minus 65 to minus 45, minus 65 to minus 45. Now, when it goes beyond that, then you can have a spike potential. So the question is, when does it go above threshold, and why does it not go above threshold at times? So for us to understand that, I will take you back to some other slides. So here. So I've talked about the basic electrical rhythm, and this is the rhythm of the membrane potential. And it starts with the interstitial cell of Cajal. And now, I wanted to show you that this one fluctuates between minus 65, minus 45. I told you that. So I want to see a graphical representation. I'll come to this slide later. I want to show you this graphical representation. Actually, this is in the this is in textbook of medical physiology. You can get that diagram. So basically, let's block that. So if there is no activity, in the GIT, you find that we have that inherent electrical rhythm where the membrane potential is rising and falling, and you can see that it is right between those values that we mentioned. Now, there are situations that can arise and it will move the whole of this block downwards. So let me see. Can you see this wave? All of it, the way it is moved downwards. So if I were to move this one in that direction, and the amplitude still remains the same, it means that now I will be fluctuating. I'll be fluctuating this wave, but at a lower position. So if it is brought downwards, it will be fluctuating at that level, maintaining the same amplitude. Now, in most cases, at the peak is where you can hit threshold. If the peak crosses the threshold level, then you can have a spike potential. If you move the whole graph downwards, it means for you to reach this threshold, this is the threshold value. For you to reach that threshold value, you have increased the stimulation you require. For you to move from this peak to threshold, you need more stimulus. Whenever you need more stimulus, it means what you have actually done is inhibited any contraction activity that is likely to happen. So what you're doing in essence in the digestive system, you are inhibiting any potential movement within the GIT by moving this graph down. There are another situation that can come. Instead of moving it down, you move it up, the whole thing. So when you raise it up, it means that now the fluctuation will be at a higher level. So if I have moved it up, now I'll be fluctuating at that point. Now when that happens, it means at one time, this peak is above the threshold. And so whenever it is above the threshold, you are going to see spike potentials. And then when you repolarize again, it is still above threshold, you get another spike potential. Again, another one follows. As long as it stays above, you have those spike potentials. Whenever we have a spike potential, if you look at the muscular activity, if they say, for example, I draw a line here for the muscular activity, as long as it is below threshold, there is no contraction of muscle. However, as long as I have a spike potential, I start seeing contraction of the smooth muscle. Now you can see the relationship between the spikes and that. Now for you to move this fluctuating graph up, you need a stimulating input that will move it down. For you to move it down, then you need an inhibitory input that will move the whole graph down. So in one case, you're going to put something there that will cause the whole wave to be lifted up, or whatever input you put there, it will cause the whole wave to be moved down. So for that reason, you can see in this other part, so if I were to section this place, in this case, you have a stimulating input, and when you stimulate, you find that the value goes above threshold and you start seeing the spike potentials. So what could be the cause of stimulation? It has been listed here. Among them, it could be neurotransmitters. You could stimulate using the parasympathetic system, release acetylcholine. Upon binding of acetylcholine, it will raise the wave, and therefore when the BER reaches threshold, you initiate spike potentials. On the other hand, even stretch, for instance, presence of food within the lumen will cause stretch, stimulating the stretch receptors, and from there you start seeing you can have not stretch receptors, stretch of the smooth muscles, and because of the stretch you allow a lot of calcium to flow into the smooth muscles, and therefore you end up having the spike potential. Now on the other hand, we can also have drugs. There are some drugs that you can give that will cause the wave to move closer to the threshold potential. Now, there are other drugs that can cause inhibition, and when you have inhibition, it's when you move it further away from the threshold, and this is when you are causing hyperpolarization. And among them is when you give a drug, something like norepinephrine, and you give some other drugs. We talk of the sympathetics. These are the ones which are similar to the sympathetic neurotransmitters. So all these will cause inhibition. Now at this point, before I move on, I want to ask if there is anybody who has a question. Interstitial cells of Cahar, depolarization, relationship to spike potential of the smooth muscle, and relationship to contraction. Any question? Cecilia?
Yes.
Cecilia?
Yes.
Are you with me?
Yes, I am.
Were you able to follow all that I was saying? Or at
Yes.
what point are you lost? Cecilia?
Yes.
At what point did I lose you?
The hyperpolarization part.
Okay. Anybody else who is lost at that point? I want to see how far back I can move back in my explanation. Hyperpolarization. Remember, you're looking at this with So you have a cell, and this cell is allowing calcium to flow in. It is leaking. So the inside is becoming positive. And as the inside becomes positive, if I were to draw a graph, it is slowly going up. And then we have a repolarization phase where it goes down. And then it goes up again. So this sets the basic electrical rhythm. Now, if I were to stretch this cell, and we say that stretch is one of the stimulus to allow calcium inflow. So when I stretch this cell, I'll have more calcium going in. And therefore, even at the lowest level, I have more positive insight. I don't know whether you captured that well. So remember, if the level of positive here, when you are repolarizing, you are starting from minus, say, 60. Now, when you have massive inflow of calcium due to stretch, even when you are at the lowest level, this minus 60 has been raised to say minus 50 because of the presence of the calcium that came in. So this lowest level happens to be at that point. But the channels that were there are still the same. So the rate of depolarization is still the same, and the rate of repolarization is still the same. But then when it goes down, because of the calcium that you are already allowed to be in, it doesn't go back to where it was. It is at that point. So now the wave has moved higher. And this is because you have allowed calcium, or acetylcholine has bound and opened calcium channels or sodium channels, causing the inside to be more positive. On the other hand, if I were to cause inhibition, it means I will move the threshold value here. I'll make it more negative. So instead of at the baseline here, instead of it remaining at minus 60, I will make it more negative here. It now comes to minus 70, the lowest point. But if that is my start point, my rate of repolarization is the same. Depolarization is the same. So this wave will have moved to a lower position. For that reason, if this was the normal and you have moved it to that area, and I want to cause a spike potential, it means at the peak, because spike potentials occur at the peak. At that peak, to move it to threshold, I need a stronger stimulus. So the whole wave has been hyperpolarized, and because it is hyperpolarized, it is inhibited. You need a stronger stimulation to initiate a spike potential. I hope that clarifies. Axilla. Axilla, are you there? Is Axilla in?
She's
Yeah, she
actually in
typed.
chat.
Okay. So now, let me move to the next one, because at this rate I will not finish motility. It's 12:04. So I will move to the next one. I hope that you are able, if I were to ask you to write about interstitial cell of Cahers, you'll be able to tell me what their functions are, where they are found, what they do, what their relationship is to the smooth muscle. Now, you find that these interstitial cells of Cahers, we find some within the stomach region, the duodenum, we find them scattered all over. But their rate of depolarization differs according to the area where we are. So that in the stomach, you find that the pace at which it sets off those slow waves is slower. When we go to the duodenum, you find that the frequency is much higher. When we go to the jejunum, again, their frequency is different. So depending on which area you are within the GIT, this BER or basic electrical rhythms will differ by giving the different number of waves within a given period of time. So for that, you find that this has been worked out, and that's why I said I'll go back to this slide. So in this slide, I wanted you to note that within the stomach, we only have about 3 of those in a minute. Go to the duodenum, they increase to about 12. In fact, this is where you have the highest number. And then when you go to the jejunum, ileum, it becomes slower. And again, by the time you go to the large intestine, it is very small. I mean, they are very few. So the numbers will differ according to where you are. Now you find that these kind of BERs are the ones which initiate the contraction movements within these regions. And usually this happens in the presence of food. So in the shall we say, in the feeding state, when you have food, then these rhythms can actually even change. They can even become higher. But when you have less food, you find that they are slower. So what I'm saying is that although I've said that there are 3 per minute, you may find that when you have food, you release gastrin and then you start stimulating more activity in the stomach. This number can rise. When you are in the duodenum, again, presence of food can cause these ones to increase. So the figure that we have given here is the lowest that you have. Now, this will set off the rate at which contractions happen within the different parts of the GIT. When we move to the fasting state, when you do not have any food at all, then you'll see that these ones go to their lowest level, and then we initiate another type of motility, which we call the MMC, the migrating motor complex, and that is the one that comes and does a sweeping movement. So I've already talked about the causes that can cause the whole of the wave to go up or down, therefore causing a change in voltage in the basal membrane potential, which is, are we going to have, remember we don't have a resting membrane potential. We have the basal, the baseline one, the lowest level. For that to rise or to become higher, what can cause the change? So this is the basal or baseline membrane potential, that one there. So this baseline membrane potential can either come to a higher level, and if it comes to a higher level, it means that now the whole graph shifts upwards. And if it goes to a lower level, so for instance here, if it goes to a level, the baseline goes to a lower level, then you find the wave goes there. So what can cause this graph to move up or down? They are the things that we have already indicated here: stretch, neurotransmitter, parasympathetic drugs or sympathetic drugs, they will move it up and down. And of course, the hormones. We talked about things like motility. Motility is going to affect motility, and therefore you find it will shift the graph up or down. So I think this graph just explained what I had told you about depolarization and repolarization. I have explained. Spike potentials I've explained. So this graph shows you the relationship between the spike potential, what we have here, and then it shows you the relationship with the tone of the muscle. So as long as the spike potential, or rather the depolarization is below threshold, then that muscle does not contract. The minute you have a spike potential, a single spike potential will cause only a slight contraction. Now, if that wave stays or remains above threshold, it initiates a second spike potential. Now, the second spike potential will cause an added effect on the initial contraction. And this added effect will change two things. One, the duration of contraction, and two, the amplitude of contraction. So in this case, it is acting like a graded potential. If you remember what we were talking about, graded potential is if it finds a spike before, you'll add on to what was there before. In this case, it is the force of contraction of the smooth muscle. If a second spike potential comes before the other one had faded, you'll add on to the previous effect on the smooth muscle. So you're going to summate. Now, if we add another spike potential, you find that there will be that continuous contraction. You can have the smooth muscle continuously becoming shorter and shorter, increasing the tension because of this stimulation. However, once the depolarization goes below threshold, that muscle relaxes. So, like I said, the force and duration of the muscle contraction will be dependent on the number of spike potentials that you have. Okay. So who can compare the spike potentials in GIT smooth muscle to those of action potentials in nerve fibers? What's the difference between the two? What's the similarity? Anybody who wants to try? You don't have to say all the answers. Just mention one thing. Do I really have people in class? Lucy Wainaina, do you want to try? Lucy Wainaina, are you there?
Yes, I'm here.
You want to try one? Just one answer. There are several.
Okay. For smooth muscles, they
Yeah.
can be summed up like they have the slow waves, but for the action potentials in nerve fibers, it's only one, like it has to reach threshold for it to be an action potential.
So for the spike potentials in the GIT muscle, you say they can be summed up. I don't know whether I got that right. In the action potential in nerve fibers, We have that one. Yeah. And the spike potential, they are usually at the peak of the BR. You don't have that hyperpolarizing phase. It never goes below. So it will be at the peak of the BR. Okay, that is one difference. Any other difference? You can see they have quite a number. From the baseline, okay, to the threshold, they are different. Okay? The fact that even within the smooth muscle you do not have a resting membrane potential. In the action potential, we usually have a resting membrane potential. So we have a baseline membrane potential for the smooth muscle. Now, the other thing is, I think that's what Lucy said, that with the spike potential, you can have summation. With the others, you cannot have. And the other one I remember mentioning, in one, we only have calcium. The other one, we have sodium causing depolarization. So remember I talked about the spike potential is initiated by calcium inflow. Now, with the action potential, we normally have sodium is the one that causes the stimulus, causes sodium channels to open. And these are the ones that will reach threshold and will open all the sodium voltage sensitive channels. With the GIT, once you reach threshold, you open both calcium and sodium channels, and this will cause massive depolarization. So we have quite a number of things that you can look at. So I want you to look at greater details on smooth muscle spike potentials, compare them to the action potential. Now, I think I will skip this because it is showing you how calcium leads to muscular contraction. And because you did this in Physiology 1332, when you are looking at smooth muscle contraction, first there is the calcium inflow, and then we have the calcium binding proteins, which is the calmodulin, from there initiating a series of events that leads to the sliding filament mechanism. So this slide was just to remind you that you need this calcium inflow. Now, initially, the calcium that comes causes depolarization. And this depolarization does not necessarily have to be the calcium that leads to contraction. So once you have that depolarization, it will lead to the spike potential, which causes more calcium to go in together with sodium, and from there, it is the one that will end up causing the sliding filament mechanism. So this slide was just a reminder of what you have. So now, once you understand that, let us look at the different processes at different places. First, we have peristalsis. These are the propulsive movements, and they usually move from the oral region towards the caudal or the arboreal region. These peristaltic waves, they usually occur they arise due to the presence of food in a certain area. So it is a reflex in response to the presence of food. So we have mechanical stimulation initiating a reflex. Usually it is local and it occurs only within the enteric nervous system. So apart from the one that starts within the esophagus, which involves the autonomic nervous system. In all these other parts, it does not involve the autonomic nervous system. It's a regional reflex. Now, the one thing that we know is that you can increase these propulsive movements by using the autonomic nervous system, whereby if you release acetylcholine, we said one of the things that the acetylcholine will do is to raise the basal membrane potential, therefore making it easy to reach the peristaltic, or rather the threshold, and therefore initiating contraction. So it will become easy to allow these peristaltic movements to occur. Now, I wanted to show you the difference here between peristaltic movement and what we call segmentation. Now, if you look at peristaltic movements, there is contraction behind the food bolus and relaxation ahead of the food bolus, forcing the food to be propelled in that direction. So if we are looking at a specific point within the GIT, at one time the food bolus was located here, and after a few seconds you find the food is located in the onward direction. So that's why we are saying it is propulsive, it is pushing the food. Now, when it comes to the segmentation or mixing movements, these are usually found within the small intestines. Now, you'll find that if we're looking at the same position of the GIT, so we have position A here, and you have position B here. So when A and B contract, you find that you will have a bulge here of the contents. When you contract these two areas, most of the contents will be forced to this middle part. And if, say, for example, this area is C and it is contracting at the same time as A and B, you find that contents will be forced in the place that is relaxed. Now, immediately after you contract A, B, and C region, the reflex will be to relax A so that this one becomes relaxed, relax B, and relax C. And immediately what will follow is the place that was relaxed here, let's call it number 1, and the place that was relaxed before, which is number 2, becomes contracted. This place contracts. And when it contracts, it will push the contents to where B was. It will push the contents to where A was, and it will push the contents to where C was. So, in this second diagram is when we have now number 1 has contracted, and number 2 has contracted, and B has relaxed. So now, if we had contents that were colored, you find that now when you contract one, because some of the contents here were yellow, others were green. Now when we contract, some of them will be pushed back, others will be pushed to the other side. And because of that, it is like taking a bottle and shaking it. The contents are mixing. So when you follow a second contraction of A, B, C, now the contents will be completely mixed. So what is happening is that you are alternating A, B, C contracts while 1 and 2 relaxes. In the next wave, A, B, C will relax while 1 and 2 contract. How many have I lost in the segmentation process? I have lost one person. So I don't know whether I should block one of these diagrams. If I block this diagram and we only focus on this one. So we have within this pipe, let's say it's a tube and it has contents within the lumen. And these contents are well demarcated in color. So now I mark one point and I put it A. I mark another circumference, I put it B, and I mark another place, I put it C. At the same time, I contract all those points A, B, C at the same time. The force of making the lumen so small here will force the contents to move away from B, but at the same time I have contracted A, so even the contents here are moving away from A. So they are being forced to come to this area. So it's like you're having Fanta on one side and Coke on the other side, and you squeeze the two bottles. Where the two liquids come and hit against one another is where you don't have a contraction. And if I mark that area as 1 and 2, so you can imagine I have Coke here and I have Fanta here. One is dark brown and the other one is orange. If I squeeze on B, it will force the Coke to move in one direction and the other one to move in the opposite direction, so that I have very little content here. At the same time, I have done that to that area, which is holding Fanta. So in the area where I have not squeezed the two, you'll find that the two forces of the sodas will hit against each other and they are going to cause mixing at that area. Immediately after I contract, I'm going to relax this place. And when I relax, immediately what I'm going to do at those points where I had contracted, remember I had contracted B. I'm still at position B. Now I relax B. And at the same time, I relax A. And at the same time, I relax C. But at the same time, concurrently, I contract where I had marked 1 and where I had marked 2. So remember, initially, the Fanta and the Coke had hit one another, and immediately once they hit one another, I contract that region. It will force a mixture of the Coke and Fanta to move back and to move in this direction. So what I will have done is now mixed the contents. Are we okay now, Lucy? I think I had...
Yes, but I have a question.
Yes.
Okay, we have said peristaltic, it moves into the forward direction.
For now, forget peristalsis. I'm talking about the mixing.
Okay.
So when we are talking of peristalsis, it is different from segmentation. So this on your right is segmentation, on your left is peristalsis. So when I was discussing peristalsis, I did not have segmentation. This is now propelling or moving the food in one direction. That is peristalsis. Now, when I start having segmentation, I totally ignore peristalsis. So now in segmentation, I have segments. In fact, if you look at the GIT, it looks like segments of portions, so that we say it is segmented. If I look at that GIT, the small intestine, it will be having bumps. And because it is having bumps, it looks like it is segmented. That's why it is said to be segmented, or segmentation movements. So is it clear?
Yeah, it's clear, but can we say that segmentation causes the maybe diseases like the acid reflux?
Oh, we have not talked. We'll talk about reflux later. Let me first go through all those methods of movement, and maybe it's already time. I want to show you something. Just give me. I don't know whether it will have audio, but let me just try and see. Okay, so now when you're given there's some barium salt that you can be given to swallow. It is liquid, but barium salt, the fact that it has barium, it will not allow the X-ray to pass through. Oh dear, there is so much background noise here. So what will happen is when you swallow, it goes all the way to the stomach, and then you can follow what is happening within the GLT by looking at that barium medium that you have swallowed. So what I have here, it is barium that has settled within the stomach, and you can see the activity that is going to happen. So I want you to see the type of contraction movements that you see within the GIT. as it moves along. Are you able to hear me? Hello?
Now we can hear you.
Okay, are you able to see my arrow?
Yeah.
So you can see what I'm talking about, the mixing. It moves to and fro. If you look at the point of contraction at one time is different from the second point of contraction. So just observe for the swinging of this barium at that area. Okay. You can see the mixing movements. So I think because on the interest of time, I'll stop it. You'll watch it at your own time. But what I wanted to do is to show you how you can have those complexes, how they can move. I don't know whether that now makes it clearer when you see those mixing movements as compared to the peristaltic movement. So they say that in the small intestine we have both peristaltic movements and the mixing movements. So what you could see in that video, you could see the barium moving to and fro, to and fro. It is shaking, and then at one time a wave comes and pushes it and propels it in the forward direction. So that's the kind of movement you're going to see in the small intestines. So I think I will stop at that. I will stop at that so that I don't start on the migrating motor complex. We'll deal with that later. So we are now in agreement that we shall discuss the pre-lab session. We'll talk about the pre-lab session on Tuesday afternoon, which means that you cannot have a lab session next week. Yeah, let's keep next week for the lab session so that I can give the pre-lab on Tuesday. So with that, I'll take attendance. Quickly, I will start with B today. So today is week 4. Justin Malu, is Justin there? Or Justin doesn't even know we have online. Sarah,
Present.
Lucy Mogore,
Present.
Lorraine,
Present. Present.
Axila. Axila, is she still having a problem?
It's typing the
Present.
Okay, Omenya,
Present.
Charity,
Present.
Prudence,
Present.
Mutinda, Philip,